We demonstrate the biology of DNA replication, leading to bacterial cell division in a gram-positive bacteria. The DNA is shown as a circular. DNA replication requires that the two strands of DNA, known as helicases, break the hydrogen bonds between the bases in the two DNA strands, unwind the strands from each other, and replication of DNA are known as replication forks. The enzyme's DNA polymerase then moves along each strand of DNA behind each replication fork, synthesizing new DNA strands, in red, complementary to the original ones. As the replication forks move forward, positive, which is also known as topoisomerase II, is responsible for removing the positive superhelical twists so that DNA replication. Quinolone antibiotics, shown here, are synthetic molecules. The term fluoroquinolones, inhibiting the activity of both the DNA gyrase and, for most gram-negative bacteria, DNA gyrase is the primary fluoroquinolone target. Quinolones appear to stabilize the enzyme DNA complexes, which in turn results in breaks in the DNA that are fatal to the bacterium. A second mechanism of fluoroquinolone action is shown here. With some exceptions, topoisomerase, the final result on the bacteria, however, is the same. Bacterial replication is disrupted and the bacterium breaks apart. The relative potency of different fluoroquinolone antibiotics, and thus their spectrum of activity, is dependent in part on their affinity for either DNA gyrase or topoisomerase IV, or both. The DNA is shown as a DNA. In order for the stranded DNA molecule must first unwind and separate in the read. Only one strand of the DNA serves as a template for this process known as transcription. Transcription results in the formation of messenger RNA, mRNA, which is mRNA is complete. It will detach from the DNA template and in turn become attached to ribosomes. Synthesis of the polypeptide chain begins. This step involves the aligning of the sequence of the tRNA, which, when aligned along the mRNA and ribosome, join together to form the ribosome will continue to add amino acids to the growing polypeptide chain until it reaches a point along the end. At this point, it releases the finished protein. and pili. In gram-negative bacteria, the gap between the cell membrane and the cell wall is known and is attached. Peptidoglycan, the most important component of the cell wall, is a polymer made of N-acetylmuramic acid alternating with N-acetylglucosamine, which are cross-linked by chains of 4 amino acids. The function of the bacterial cell wall is to maintain the characteristic shape of the organism and to prevent osmosis. Synthesis of the peptidoglycan and ultimately is the addition of 5 amino acids to N-acetylmuramic acid. Next, N-acetylglucosamine is added to the N-acetylmuramic acid to form a precursor of peptidoglycan. This peptidoglycan precursor is then transported across the cell membrane to a cell wall acceptor in the periplasm. Once in the periplasm, the peptidoglycan precursors bind to cell wall acceptors and undergo extensive cross-linking. 2 major enzymes are involved in cross-linking: transpeptidase and D-alanine carboxypeptidase. These enzymes are also known as penicillin-binding proteins because of their ability to bind to penicillins and cephalosporins. Eventually, several layers of peptidoglycan are formed, all of which are cross-linked to create the cell wall.
The basic building blocks for the peptidoglycan backbone, N-acetylglucosamine and N-acetylmuramic acid, are synthesized in the cell cytoplasm. A chain of amino acids, usually 5, is then added to the N-acetylmuramic acid. Once the building blocks have passed through the membrane, they are enzymatically added to the growing peptidoglycan chain. Next, the transpeptidase enzyme complex completes the synthesis of the peptidoglycan macromolecule by cross-linking the individual strands. The general process of peptidoglycan completion proceeds as follows. First, the terminal.
This is an antibiotic. We all know it. What a great invention. Saving lives since 1928. Patients love it because they trust it. Doctors like it because they know it works and because their patients trust it.
Beta-lactam antibiotics include all penicillins and cephalosporins that contain peptidoglycans. Beta-lactams interfere with cross-linking by binding to transpeptidase and D-alanine carboxypeptidase enzymes, thus preventing bacterial is damaged. Gram-positive bacteria have a high internal osmotic pressure. Without a normal rigid